Hey everyone, and welcome to our podcast series straight here from the Charles Darwin Research Station, exploring stories of science and wonder in the Galápagos Islands. I'm Amber Tonti-Lamotte. I am the Director of Marketing and Communications, and I'm super happy to have today Sonia Kleindorfer here with us. She's a collaborating and visiting scientist who has been coming here for over 20 years, studying endemic land birds of the Galápagos. She's been doing some incredible work, is just fresh from the field back, and sitting with us today to tell us about what she's been doing. So Sonia, welcome. It's really great to have you today. So maybe can you tell us a little bit about you, what you do?
Yeah, it's great to be here. Thanks for having me. Animal behavior is my passion, and I study the behavior of birds to use insights gained from their behavior to foster conservation outcomes. So the more we know about them, the more we can help them with targeted interventions. But I'm basically fascinated by animal behavior. I'm a professor of biology at the University of Vienna and director of the Konrad Lorenz Research Center for Behavior and Cognition. And I've been working on the Galápagos since 2000, and specifically on Floreana since 2004, with a focus on Darwin's finches, and most recently the short-eared owl.
Oh, wow.
Yeah.
And what's happening in Floreana, actually? Can you put things into context a little bit for us? How does your work fit into that?
Floreana is undergoing the largest ecological restoration project that I've certainly heard of or been involved in. It's a huge privilege. So many organizations working together to achieve outcomes to restore the island. What I can say over the past 20 years is that there has been a decline in the populations there. So the Darwin's finch populations have not been doing well. We know that 5 of the 10 original Darwin's finch species are extinct on Floreana Island in the last 100 years. And the populations really have been declining, and it's a situation of concern. At the same time, we noticed that the rat population was increasing. So we've been monitoring the rats using rat tunnels, for example, baited with peanut butter. And when they come in to eat the peanut butter, and they leave their footprints. And so we know that the rat numbers have increased. And I know this personally because when I went to check the Darwin's finch nests, you put your hand in the nest to count the eggs or the chicks, for example, and out would peer a rat. And they would be climbing trees everywhere, and we'd sleep under domes in our beds and have rats crawling on the outside. And so the locals, said, well, we know how to deal with rats, and so they brought cats in, which is fair enough. And then over the years, we used to have lots of lava lizards walking around and lots of marine iguana walking around. And from 2016 onwards, we're basically cats hunting and rats crawling all over us and every finch nest. So that's where, the Galápagos National Park had obviously been designing, planning, working with the community a few years previously to eradicate the rats, and implemented the eradication program in 2023. And so because we have these 20 years of baseline data, we can compare the impact of the eradication now on the Darwin's finch recovery, or at least their behavior in response to the massive reduction in rats and cats. Yeah.
Wow. So just for the context for our listeners, the Floreana Ecological Restoration Project is a massive undertaking. I think it started 12 or 15 years ago. Huge planning element. And like you said, in 2023, we had the first control or the eradication activities that took place, specifically targeted at rodents and those feral cats, right? There's also a component of habitat restoration, and ultimately the idea is to reintroduce about 12 locally extinct species, right? And a lot of them are land birds, hence why your work is so relevant. And I think one of the birds actually, and not the reintroduced ones, but one of the birds that's been most affected, I think, by the rodents and the cats in particular, was the medium tree finch, right? Can you tell us a bit about that?
Yeah. The medium tree finch is critically endangered, and it only occurs on Floreana Island, so it's an island endemic.
Okay.
And Darwin's finches have this really cool feature, which is that they increase the proportion of black plumage in their head with every year of molt. And they get completely black heads by the time they're 6 years old. As yearling birds, they're gray. And they can already reproduce as yearlings, which means they're one year old. And what I had noticed over, these decades was that the population was really old. It was like all these black-headed males, and there was hardly any recruitment, which means that they weren't having reproductive success. Now, we know this because we would go out and find the nests and monitor the nests and then check if there were any fledglings. And on average, for 20 years, we had one nest of all the nests that we monitored with fledglings. I mean, that was incredibly low reproductive success. Yeah. So we had this really aging population just 2 weeks ago because we color band the birds and we put unique color combinations in a unique ring on their leg. We recaptured one who was 18 years old. Wow. So these small Darwin's finches, you would never imagine that they live that long, but they live to 18 years at least, we now know. Anyway, so I was watching this really aging population. I was very concerned because when you go and then you count every year and you hear, oh, but they're still there. But if they're not reproducing, then at some point they're all going to die off. So that's why we were very concerned. Yeah. And we have wonderful news from this year. I'm really excited by what I just saw. I've just come back from 2 months on Floreana. Yeah. And we did the program that we've been doing for 20 years, which is to find the nest and monitor the nesting outcome in this critically endangered species. And there were now, of course, we need to analyze the data, but my first impression is that half the population of the birds that we misnetted, and certainly of those that we observed, are yearling males. And I've never seen that in 20 years, which means that they had this surge in reproductive success, which is amazing. And then from the nesting success, previous years we had one that produced offspring, one, and now we're monitoring 15, and we already had 7 fledge. I mean, so yeah, it's just a huge increase in reproductive success.
Wow, and that's directly related, in your opinion, to the rodent control and the cat control. Yep. That's incredible.
Yep.
Wow. And so you're doing also other actions in order to protect them, because surely the rodents and the cats aren't the only threat that they're facing.
No, there's this creature called the avian vampire fly, which is the biggest threat to all Galapagos land birds. Right. It's a fly that's about the size of a house fly. It's vegetarian. It was accidentally introduced and first discovered in Darwin's finch nest in 1997. And in the past, 2 decades, the first decade of its impact was really killing the finches very young. And then in the second decade, because the finches were dying so young, it feeds on the blood of nestling birds inside the nest and comes out at night, hence avian vampire fly. It's a blood-sucking fly active at night, but only of the chicks. So the adult finches don't have the parasite, but so the fly enters the nest, lays eggs. The eggs hatch, turn into larvae that are blood-sucking and crawl up and suck the blood of these chicks. And because the impact was so tragic, all of these chicks were dying really young. And as a result of them dying very young, the larvae pupated sooner. And from a smaller pupa, you get a smaller fly. And from a smaller fly, you get fewer eggs per female. And so what I just saw last week is the pupae are now tiny in comparison to what they used to be. So this is coevolution in action before our eyes, right? There's massive selection for the fly to become smaller because it's not in the fly's interest to kill its food source, right? So it wants to keep the chicks alive long enough that it can feed. But Phalaenopsis is still a problem. And so what we've done to assist is manually spray nests with an insect growth inhibitor. And this has been worked out together with Birgit Fessel and colleagues from Charles Darwin Foundation. Over the years with Sarah Knuti, there are lots of teams that are working on mitigating the impacts of the fly. And one thing that we did was to manually spray the nest with insect growth inhibitor, and it's sufficient and the nests are at like 6 meter high, 8 meter. So you have this massive pole and you're spraying the nest on the outside, and that is sufficient to kill all the parasites and or prevent the female from ovipositing. So if she oviposits, then the growth inhibitor means that the eggs don't develop. So that, we think, really helped the medium tree finch, because we did that at 20 nests last year. And we know that they produced fledglings as a result of that intervention. This year we did not do that for various reasons, but we're continuing with the fly feather dispenser materials, which is placement of these materials where the finches can go collect feathers, collect cotton that's treated with this permacap insect growth inhibitor, bring it back to their nest, and they self-fumigate right? So
that's smart.
And so in that way, it's been a real partnership with so many people over so many years to develop these methods to help the finches overcome this other threat.
Yeah, and this is a technique that's used on other islands, I understand. I mean, I know our team is working actively here with the vermilion flycatcher, also using these self-fumigating stations, they call them. And it's pretty exciting when you see the little birds and they really help themselves. So you've just come back, right? You said from Floreana. What else did you observe? So the medium tree finch is one thing, but did other birds benefit from the control of the invasive species?
Yeah. So we do regular transect surveys and have been keeping an eye on the Galápagos dove, for example, and the dark-billed cuckoo. And both of these species are also, winners of this massively reduced predator situation. It's incredible. So in previous years, we had like one sighting of a dove every now and then. And this year we had 70. I mean, 70. And we accidentally caught them, as bycatch when we were trying to misnet Darwin's finches. Extraordinary numbers. And the dark-billed cuckoo, again, we had, one sighting per year. And I think we have 54 sightings and 4, accidental bycatches. I mean, you go to check the net for Darwin's finches, and there's a dark-billed cuckoo that before was a rare species, and now you see it everywhere. Like, it's incredible. And the community members on Floreana are really happy because a lot of the elders have these fond memories of the doves when they were children. And it's really nice to hear them reminisce about, their childhood and that reconnection with the way it used to be.
That's lovely. And the dark-billed cuckoo, that one is actually really shy, isn't it? It's actually really hard to see.
But I mean, we were, every single day I saw, when I was doing the transects walk, 6 to 7, it was crazy. It was really crazy.
That's really cool. But so again, the control actions is having great effects. I mean, observations seem to say it's having great impact on endemic bird population. That's great. But it's not just the endemic ones that are benefiting, right? So what did you observe, for instance, with the smooth-billed ani? What's happening there?
So the smooth-billed ani is doing very well as well, and it's an introduced species. Perhaps to add, our team, led by Petra Sumasgutna, the owl team, so we have a Darwin's finch component and a short-eared owl component. The short-eared owl team went and have been putting GPS tags on the short-eared owl to understand the movement of the only native predator on Floreana, of Darwin's finches, I should say. And so we understand the movement, and the short-eared owl is actually an island hopper, so they can go to various islands much more frequently than we previously thought. Anyway, they caught the 65 owls from Floreana Island to keep them in safeguarding, because the owls would eat the poisoned rats, and in order to keep them safe, they were brought to Santa Cruz, where they are doing very fine. In captivity. They've really adjusted and settled in well, and everybody agrees that they're in absolute top condition, which is wonderful. But the absence of the owls, and that's why I wanted to tell that before I get to the ani, the absence of the owls created a predator, like an avian predator-free zone that the ani just moved right into. And I know it's maybe, like over-interpreting what we saw, but when the owls were there, the ani, was present, but not in a cocky kind of way. And now, the ani, they just have this attitude that they're like king of the heap, king of the pile. And they're just kind of like bullies. They like go up, and I do playback where I play like Darwin's finch songs. And I always get like ani coming now to look, and that never used to happen. So the ani are kind of, they feed on chicks, Darwin's finch chicks. So they're now the new top predator of Darwin's finch chicks, and it's an introduced species. And because nothing is controlling them, and the finches are doing so well, the ani is now also doing very well. So so they're a winner as well in this story.
It's tricky, right? Because when you remove one thing, another one comes in. Like, it's always a delicate balance. One species comes out, then another one starts taking. So how do you factor in, or how do you manage such uncertainty? Or maybe it was all planned. Maybe you already knew this might happen. How do you deal with that in such a restoration endeavor as this one?
So the vision is to reintroduce 12 locally extinct species, and one of those is the Galápagos hawk. And the Galápagos hawk feeds on ani. So I have great hopes that when the Galapagos hawk is introduced, that it will find an abundance of resources to sustain it and will naturally control the ani population. So the two species that I'm most excited about for the reintroduction, I should say the Darwin's finches, but really the two that I'm most excited about are the tortoise, because it's at the basic level of the trophic chain. It's a herbivore, so a plant eater, and the Galapagos hawk, which is a top predator. And so if you have these two ends of a trophic system, with the tortoise and the Galapagos hawk, they're really going to manage the in-between layers. Having top predators stabilizes ecosystems, so I have great hopes that the introduction of the Galapagos hawk will stabilize the ecosystem. The Galapagos tortoise is going to be very important for the habitat ecological restoration, right? A really key part in the Floreana Ecological Restoration Project, because the Scalesia is under huge threat. There are lots of invasive species, and the tortoise is going to really open up areas and allow colonist species like Scalesia, to have germination success. And so areas where there's the tortoise, there are lots of Scalesia, and that will sustain the Darwin's finches. And the hawk will really manage the ani, also the dove population, so it doesn't get out of control. Because there are only so many seeds for everyone. There's only so much food, right? Yeah. And so you need to have all these trophic levels in an ecosystem that balance out. And so the National Park, with all of the partners, has thought this through. And that's what these 12 reintroductions will help to achieve. Yeah.
It's incredible. Again, it's coming back to these kind of puzzle pieces, and it all kind of depends on one another. Yeah. So you touched on habitat restoration, and so I understand. So the medium tree finch, it lives in the Scalesia habitat, right? And so how did you observe? How's that habitat doing? What do you see there?
I would say I am concerned about the Scalesia forest. I mean, it's one of the most precious endemic forest types in the world, and only 1% of it remains on Galapagos, right?
In the whole archipelago, this is a stat.
That's right. And so Floreana has a relatively, large Scalesia forest, but over the last 10 years, several introduced vine species have just been toppling the trees. And one of them, I can never remember it, is it the rosary vine?
The one with the little red beads?
Exactly. Is a hugely toxic and destructive force toppling the Scalesia forest. So we really need focus on the Scalesia health. I have great hopes that the tortoise will, make sure that the area is open and that the Scalesia can thrive. We have concurrently, in addition to plant monitoring, invertebrate monitoring as well, together with Heinke Jäger and the entomology group here, really trying to make sure that there are enough invertebrates across the year for all the species reintroduction. So, getting the numbers right is going to be important, but we have information about all of the component pieces, and it's all being put together now. So yes, the habitat ecological restoration, the plant restoration, the invertebrate diversity and abundance are key components as well in this story.
Yeah, that's super interesting, and that's something that the foundation is going to be looking at specifically in that project, mass habitat restoration. And so I just wanted to jump back to the Galapagos hawk very quickly, because earlier you said that the short-eared owl is an island hopper. That was new to me. I did not know this. I thought, well, first of all, I've never seen it crossing between islands, but you would think. So the Galapagos hawk isn't? Because it's present on other islands. So that's not an island hopper.
Oh, we'll know when we put the GPS tags on it, yes.
Okay. Because, I was wondering, because given it's one of the species out there that's no longer, I thought maybe. Interesting. Yeah. So you're going to put GPSs on the hawks down the line. That's really cool.
And it was only with the GPS tags that we realized just how much island hopping the owls do.
Right.
Like in this last month, we had a few that one went to Isabela and Santa Cruz and back to Floreana. Another one went to Santa Fe twice and Santa Cruz and then back to Floreana. So
I'm gonna spend the weekend over there.
They really do travel much more than we think. Yeah.
So now going into like funny things that birds do. So you said you study the behaviors of birds, and I believe you also study the bioacoustics of birds, right? The types of songs. So tell us a little bit about the work. Like you study the songs of birds. How does that work? What is that all about?
So Darwin's finches are a great group. The sister species to the ancestor is the warbler finch. And if you listen to the warbler finch song, it's got, buzzes and whistles and syllables, and it's got like everything in it. And then as you look at the ground finches, they have certain components of the warbler finch song, and the tree finches have others. So the tree finch song is composed of syllables, cha 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 cha, or twee 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 twee, buzzes, bzzz, and whistles. So they put these component pieces together. So we're deconstructing each of these and trying to understand them. But to get to the syllables, each Darwin's finch, tree finch, produces a song with a single syllable type.
Okay.
And they learn it apparently from their father. However, that is now being thrown into question on the Floreana system based on some observations, which I'll happily share with you. So a finch produces the same syllable for its entire life. Cha 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 cha, or chwee 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 chwee, or chwee chwee chwee. So these are all syllable types. And what we do is we record them because they're color banded.
Yep.
So we can record the same male across years. We know that they produce the same syllable type for their lifetime, which can be 18 years, as we just discovered.
Yeah.
So they have one syllable type they sing. At a population level, we know that the small tree finch, for example, has, in the previous years, 10 different syllable types. Some are common in some years and some are rare. The common syllable types are not necessarily common every single year, so there's flux. And so a syllable type that is rare, let's say in 1 year somebody innovates and has a new syllable type like chwa, 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 chwa. And then a few years later, 40% is singing that syllable type. So you can look at the emergence, the innovation, a cultural innovation, because it's learned and passed down across generations, and the spread of that cultural innovation in a group. And so we've been doing that for every Darwin's finch species on Floreana. And another really surprising discovery that we have yet to analyze and fully explore, but the 3 bioacoustics members of the team are all in agreement that this year we had an explosion of cultural innovation, an explosion of new syllable types and new combinations of syllables that we've never heard in the previous 20 years. And that really got me thinking. I was like, why this year is there so much cultural innovation just in the absence of predation risk, right? When the environment is safe, why do animals innovate then? And it kind of stopped me and it got me thinking. I was like, well, actually, it's probably reasonable and to be expected that you would innovate when there is no cost to explore, right? And so when you remove the predation risk, the cost of taking risks decreases. You're not going to get killed if you venture to the periphery. So I think when risk is decreased, so in times of plenty, in times of safety, that's when you're more likely to innovate. And that's certainly what we seem to be seeing. The other observation, though, is that we had so many fledglings this year, like so many surviving youngsters.
Wow.
And we observed these flocks of fledglings, teenagers, teenage hangouts that before we had never seen, because when a finch fledges, they're not good flyers, and so it's just easy for the cat and the rat to eat them. And they really don't look like they can fly very well. They cannot fly very well. But now everyone's surviving, and what we saw were these teenage groups, mixed species, where all the juveniles come together. And so we're thinking, well, maybe it's like that assortment of the juveniles, the group size is much bigger, there are many species, and so maybe they're more open to learning from each other. And the outcome of that, whatever the mechanism is, we'll be studying it, but whatever the mechanism is, that in terms of the innovation, the novel song types, we have so many. We have, for the first time ever, 2 syllable types composed as a new syllable. We have double buzzers. I haven't spoken about the buzz, but really a lot of innovation happening. And it just is right now for the first time after 20 years. And we wouldn't know that if we didn't have the 20 years of baseline data. So it's really exciting.
That's fascinating. Yeah. It's so funny how they just become more creative because they feel maybe more relaxed. And
or perhaps, so we study personality. And we measure personality in Darwin's finches. We have There are 5 axes of personality, which is boldness, aggressiveness, sociability, activity, and exploration.
Okay.
And we have repeatability. So in order to qualify as a personality trait, it has to be repeatable. So across years, if I put you in a novel arena, which we have with the finches, and we film them, some will explore every corner and others will stay in one spot. The one that stays in one spot is not exploratory. In a novel area, if you go to 12, then you have a really high score. And then we release them. It takes 5 minutes. And there's high repeatability. So if you're exploratory in one year, you're exploratory in another. And it correlates with whether you're likely or less likely to approach a novel object that we also test experimentally. So if you're open to novelty, right, then you're more likely to copy and innovate song syllable types. So we're going to answer or provide, or at least ask the question and have an answer as to whether there has been a change in the exploration score of the population after the eradication. Because being exploratory is risky, right? You could, while you're exploring the novel object, the owl or the rat could get you. But there are no owls or rats. And so everyone can go and explore and be open to novelty. And I think that's one of the mechanistic pathways for innovation.
Could one also be worried that they could lose the knowledge of like just being careful about their surroundings if they're too free for too long? Let's say if they don't have any predators, could they lose that ability to actually watch their backs? Yeah,
but usually you would imagine variance in the population for the trait. And so all of those that are too exploratory could be selected against when predators come back. But the shy ones or the cautious ones, they would still be in the population and they would then produce more offspring. And so you would have these shifts in those traits based on the prevailing ecological conditions.
That's incredible.
So,
that's super interesting. And it's funny because you can actually draw parallels, especially with the creativity piece of the songs with humans ultimately, right? But how does this work specifically, the song work, for instance, and the behavioral work contribute to then better conserving the birds? How does that, like in our future planning, what are we learning for the future that will help us do it better so that we can help them better protect?
For example, if you had to select a group of individuals for a translocation, you might want to select individuals that would be less exploratory and that would then, be more likely to stay in the area that you translocate them to, rather than run to the edges, and try and find all of the novelty of that environment. I mean, that's one example. So understanding personality profiles can help to translocate. You might want to select for variance. You want to have representatives of all of the personality phenotypes, and not just the ones that are easy to catch. So there are ways that this information can inform who you target for translocation. And also, in terms of the animal culture, which is defined or can be defined as learned behavior that is passed down across generations, you would need a combination of personality phenotypes, for example, to protect yourself against a predator, which would require bold individuals. So some individuals might be bold and have a function in a group of protecting others in the group. So we see that in social animals, so that some are bold and they have predator defense, so that the exploratory ones can be safe and innovate, but then you need social ones that pass the innovation on via social imitation, right? And so you really want to have a combination of the personality types to fulfill the different social roles in group living species. So there are many ways that it can be useful, and we're just beginning to tap into that.
That's incredible. That's incredible. I mean, that's just like scratching the surface, I feel. So maybe my last question I wanted to ask you is, next breeding season, what are you hoping to see there?
We're certainly going to be tracking the song syllable innovation and how that relates to personality and nesting success. We are very curious how the increased density in host nesting density might negatively be associated with a rise in the parasite abundance. So we're keeping a close eye on the avian vampire fly. But because we have our tools the tools at our disposal to manually spray and put out the self-fumigation, what did you call them?
The self-fumigation stations.
I call them feather dispensers. Right. The self-fumigation stations. That we feel that we're going to be able to manage Floreana downsii, the avian vampire fly, but we're keeping an eye on that. And of course, we're very much excited about monitoring the reproductive success and looking at the age progression of these 1-year-old finches, of which there are so many this year, and looking at their development and survivorship, and hopefully track them until they're 20. Yeah.
Wow. Well, thanks again, Dr. Sonia. It was great to have you, and thank you for all these amazing insights, like literally fresh from the field. I think you came back yesterday. Yep. So thanks again, and we'll see you then at the next breeding season, and maybe we can do another one of these.
Would love to. Thank you.
All right. Thanks.
Bye.
For our listeners, the Floreana Ecological Restoration Project is a massive undertaking. It is led by the Galápagos National Park Directorate, the Biosecurity Agency of the Galápagos, and it is co-executed between Hokotoko Foundation, Island Conservation, and the Charles Darwin Foundation, with the support of many collaborators, such as Sonia, and is really like a fantastic and really impressive project to follow. So thank you so much for listening to us today. You can like this episode, you can follow us on social media, and we'll see you next time. Take care.